Hi students, thank you for your interest in my pilot project with Wharton Summer Research 2018. Over the next series of slides, I'm going to describe the project and how you could become involved, what your activities will be, and the purpose. The title is Immunohistologic Analyses of Excess Dietary Iron in Intestinal Tumorigenesis. I'll let you look at the abstract in detail, but let's get started. Which would you prefer to eat? A nice tasty bowl of cornflakes, or B, a steel oil tanker? Tough decision, eh? Actually, both have a similar form of iron. You see, the iron that's in our foods, like breakfast cereal, actually is the same kind of iron that's used to make steel ships. And in fact, many of the steel ships that sailed the oceans decades ago have been recycled, acid digested, and pulverized into iron, purified and sold back to breakfast cereal companies and put back into our foods. Don't be scared though, it's safe. As you see, breakfast cereal if you smash breakfast cereal and use a magnet, you can actually see the iron particles. They're coated with the yellow flakes of the cereal, the powder of the cereal, but they're actually visible to the human eye. Of course, many foods have iron: red meat, salmon, chicken, spinach, seeds and nuts, grains, different forms of iron, of course. We also get iron from dietary supplements. So why is iron important for this study? Well, dietary supplements, highly iron-fortified foods, and meat containing iron are widely consumed in the United States, often in excess. Iron plays a role in many vital functions, including growth, cellular homeostasis. But excess iron may in fact influence a number of diseases and actually promote development of a number of diseases: infections, cancers, cardiomyopathy, arthropathy, and other diseases. Over the past number of decades, last century, iron consumption has increased quite a bit in the U.S. food supply. Excess iron, in fact, is associated with a number of different diseases, as I mentioned, but let's focus in on a few here. So as you'll see on this slide, lateral sclerosis and certain types of cancers, along with other inflammatory diseases, are common. As you see on this slide, cancer types, including colorectal cancer, for both men and women, colorectal cancer is about 10% of all the cancers present. The incidence, prevalence of cancer in our society is in fact quite high. This slide shows a picture of colon cancer. You'll see the polyps and nodules. These are known as adenomas at this stage. But if they develop and spread, we could see occlusion of the intraluminal space, blockage of the intestine, and perhaps even metastases in the body. Here's a slide showing the stages of development. In this study, with excess iron, we'll be looking at the different stages of development and using microscopy to understand the slides and what's happening over time. So a little bit of background between iron and cancer. So iron necessary for cell growth, but iron depletion or excess could influence cell growth. There are a number of studies that have showed that either inhibiting iron or adding iron to cells, they'll grow in different ways and tumors may develop. Iron is required for cellular proliferation and differentiation. In fact, iron is indispensable when it comes to DNA synthesis. And getting rid of iron or doing something to inhibit its biochemistry will in fact severely alter the cell's stage and cycle. The hypothesis that iron intake was related to an increased risk for intestinal tumors in humans has been developed based on epidemiological evidence associating a high meat intake and development of colon cancer. And then few further studies show that it wasn't just the meat intake per se, but even separating out and feeding iron separately increases one risk for cancer of the colon. You may be familiar with the NHANES type studies data. This is the Nutrition and Health Examination, an Analyses of Nutrition Information Examination Survey. And looking at that data showed that over a 10-year period, people were developing at increased risk and developing cancer when they had higher transferrin saturation as compared to controls. Possible mechanisms might include pro-oxidative inflammatory effects, cell proliferation effects, and just that the unabsorbed excess iron may in fact initiate carcinogenesis because it can cause DNA alterations and catalyze pro-oxidative reactions. Here's a slide showing, I won't read this in verbatim, quite a bit on the slide I know, but it just shows some of the effects of iron, whether heme iron or non-heme, acting as a catalyst for carcinogenic compound development, lipid peroxidation, and you'll see at the bottom there the progression. On this slide, these are some of the proposed mechanisms for iron-mediated colorectal cancer. You'll see that iron can be involved in DNA damage, lipid peroxidation, oxidative stress, inflammation, and influence even the microbiota. So it's important, by determining the effects that excess iron has on intestinal tumorigenesis, more specific dietary guidelines regarding iron may be developed to modulate tumor progression. So nowadays, in terms of iron recommendations, many patients leave after treatment for cancer and they're just told that to eat a healthy diet, balanced diet. But you know, often patients think if a little is good, more is better. But that's not the case with so many things in nutrition. It's about sufficiency. Deficiency will cause risks and there's danger, but also excess. And in fact, consuming too much excess dietary iron is often problematic for many cancer patients. The purpose of this project is immunohistologic analyses to determine whether increased dietary iron modulates tumor cell growth and differentiation. In the past, we used a mouse intestinal tissue, a mouse model of intestinal tumor development. And we'll be looking at normal and cancer tissue from those previous studies done over the last couple of years. My lab has a number of tissue slides that we'll be staining and then analyzing and evaluating to look at the progression of tumor tissue in comparison to normal tissue. This slide shows, in fact, normal tissue in the purple box and tumor tissue in the red circle. And you can see that the intestine has been stained blue. You can actually see some of the villi folding over after the intestine has been cut, and then the tumor. This slide shows a group of tumors, and the mouse intestines have been cut, stained blue, opened up longitudinally, and then the tumors are measured. You can see the grid lines here. The tissue samples that we'll be working with on this project have been cross-sections of the types of tissue you're looking at now. So, in this study, students would assist in the analyses and evaluation of tissue immunohistochemistry. The H&E Ki67 beta-catenine at the bottom there indicates the types of staining for either visualization or proteins and other growth factors, cell cycle factors. Sample images. Here's the type of image that you would be looking at. And we see a cross-section here of the mouse intestinal tissue with the villi pointing upwards. Quite beautiful, if you ask me, in fact. Nice brown, purples, shades of blue, teal. Here's the cross-section of intestinal tissue showing a tumor growing, and actually, to the left, you can see it pushing the normal villi aside. Here's an image of both normal and tumor tissue, tumor on the top, adenoma tissue on the top, and for comparison. The hypothesis and purpose of this Wharton Summer Student Research Project. Hypothesis is that excess dietary iron modulates tumor formation and growth and thus influences prognosis and survivability clinically. The purpose of the project is to investigate the effect of excess dietary iron on intestinal tumorigenesis in mice using immunohistochemical analyses to measure aberrant crypt, foci development, transformation, and growth as they mature and transform into tumors. So what specific requirements are necessary for the chosen student? Well, you should have a good basic understanding of nutrition and understanding basic human mineral requirements and functions of minerals in the body, have some laboratory experience. In other words, you should be able to use a pipette to weigh, to balance, understand what basic lab equipment looks like, and even some basic microscopy would be good. Good time management and organizational skills are good for every project. Good verbal and written communication, of course, is necessary. And you should have some biological science and biochemistry background. Student outcomes. So you as a student would be able to, not just at the conclusion of this study, but I would think even earlier on after we begin, to understand and interpret immunohistochemical results as related to diet and cancer, demonstrate proficiency in using advanced microscopic techniques. Note you'll be trained and given assistance along the way for all of these. You would apply statistical approaches to process and interpret data sets and enhance oral and written reporting and presenting skills. Let's go back one, sorry. Student activities will primarily include microscopy, immunohistochemistry slide evaluation, data, statistical analyses, literature review, and writing reports. Some weeks, sometimes you'll be doing more so one versus another, but overall, these are the primary student activities. Thank you very much for your consideration, and I'm always, of course, available for questions as a faculty here in the nutrition department.